So this is our second lecture, and I just remind you that we are talking about the early questions about heredity. And in the first section, the section that we are covering, we are talking about why are children like their parents. Then we will talk about what is transmitted, if miniatures of messages, and then we will talk about if the traits are acquired or not. So let's start with this one again. And the question of today is, what are the early ideas of inheritance? And we're going to start as usually with the Greek philosophers, and then we will try to make a connection to how these Greek philosophers influence important scientists like Darwin. So I hope that today you come with some ideas of how the Greek philosophers approach these problems, and also very important, how they influenced Darwin to develop this idea of genetics in the early stages. And just to remind you, last time we talked about the scientific method, and we talked about that the scientific method is not a subject, it's a way of learning, and we talk about all these different components: observation, question, hypothesis, experiments, analysis, and conclusions. And today we will show you that at the end, Aristotle was the first one who, in addition to have observations, question, and hypothesis, he was the first one who developed experiments. And still, the scientific method is not completely developed, but that was the farthest that the Greek philosophers were able to achieve. So I'm going to give you a small question that it can help you maybe to study for the test. Try to answer these questions. In the process of science, which of these is tested? Results, observation, hypothesis, or a control group? And if you remember our previous class, we talked about what is a hypothesis, and we give you examples of hypothesis, like the light bulbs. And here we talk about, give two examples, and I show you that with experiments we can test the hypothesis. So the answer, of course, is a hypothesis. And if you want to study more in detail this topic, and you can go to the Campbell Biology. And if you don't have a Campbell Biology, you don't study biology, I will leave this link that has the scientific method chapter is available online, and you can read it and understand in more detail and do very well in the test. And in addition to the scientific method, we talk about early ideas about inheritance. So we talk about different examples of domestication. And I told you that actually selective breeding is not only in the past, but for example, we are still selecting breeding. We have been selecting breeding animals like dogs, and there is also examples of other animals like chickens that have been artificially selected for many generations. And I give you the example of the corn, and we talk about that selective breeding allowed to propagate traits while eliminate the ones that are undesirable. And we talk about all this history about selective breeding of corn and how we can use genetic tools to track the rate of mutation and how corn appear. So now let's try to answer this question that also can be in the test. So how many genes control the difference between maize and theosynthesis? And we studied that topic in the previous class. So what do you think? A, B, C, D, or E? And you can stop the video and think about it. And of course, the answer is this one. And if you remember, the way that they found the answer, it was through simple Mendelian genetics. The simple process that we use, and I just remind you, go and study those topics in the Campbell Biology, so you do very well in the class. And in their case, through those simple monohybrid, dihybrid crosses, they found that between 4 and 5 genes are necessary to produce these dramatic changes between theosynthesis and maize.